before understanding next generation sequencing, we should start with what is sequencing exactly. So as we all know that like there is DNA in our cells, and on that DNA there are nucleotide sequences that are placed there in particular fashion to differentiate one person from another, right? So in sequencing, what we are doing is we are exactly determining the order of the sequences which are there on DNA, because when we have to do some NGS analysis, so we there identify that what is difference, what sequence is difference between the normal samples and the disease samples. So it is very understand the sequence of the nucleotides. That's how sequencing comes from. So for sequencing, we definitely need some device to sequence these nucleotides. So these are the sequencers. And for that, we use next generation sequencers in when we talk about next generation sequencing. So basically, next generation sequencing devices started in 1990, even before Sanger sequencing, but it was not commercialized. By 2005, the first NGS technology, the sequencer was commercialized and came into market. So afterwards, the people, the end users were able to use these sequencers to identify the different sequencing patterns in the samples. What are the advantages of NGS over other conventional methods like before next generation sequencing? We have the first generation sequencers, including Sanger sequencing. So the advantages including earlier, it was not possible to parallelly sequence different samples, but in NGS, we have the facility to sequence different samples together. Even we sequence million of samples together. So this is one of the advantages because it will make it time effective as well as cost effective. Second is it provides higher discovery power to detect novel genes. Even we don't have the reference, even we don't know the existing sequences that how normally the sequences are in the human body. Still, it has the potential to identify the novel genes, new genes which are not yet been known even. The third is it have higher sensitivity to quantify rare variants and transcripts. So basically, there are different type of technologies inside next generation sequencing when we perform. So there is deep sequencing and deep sequencing analysis and others. So at different depth of the reads or the sequences which are present in the body, even the intensity of the sequences or the expression of the gene is not that high, still these sequencers are able to detect those genes within the samples. Additionally, it is also known as hypothesis-free approach because we don't need any prior knowledge of the sequence. As I mentioned, that we do not need any reference, for that. It will identify the sequences and the sequence orders which is available in the sample so that we can compare it afterwards as well.